Okay, as I said at the end of the last podcast, I was going to have all inversions and translocations on the same podcast, but I wanted to break it up because it was getting to be too big of a podcast and it would have had problems uploading. So in this podcast, we're going to focus on translocations. Let's go ahead and get started. All right, so let's start talking about these translocations, and I'm going to begin by defining the difference between a non-reciprocal translocation and then a reciprocal. Sometimes the non-reciprocal ones are called simple translocations. So what happens with a simple or non-reciprocal translocation, you're going to have a chromosome like so, and we'll put our AB there like we have been doing, C, D, E, F, and then let's draw another chromosome like so, and we'll put genes G, H, I, J on this one. In a non-reciprocal translocation, we'll have part of a chromosome from one chromosome move to another chromosome. And now it's important to remember when we're talking about translocations, we're talking about movements between non-homologous chromosomes. Okay, so GH here, for instance, is going to move to this chromosome. So you're going to end up with something like this, where you have GH here getting moved in front of the AB. Then you're still going to have C, D, E, and F. And then you're going to have this smaller chromosome here that just has I, J. These are less common in healthy cells. We do see these kinds of translocations occurring in cancer cells. We're going to spend most of our time talking about reciprocal translocations. So let's draw our two chromosomes again. A, B, C, D, E, and F. And then we'll draw our smaller chromosome here. G, H, I, J. A reciprocal translocation would be when one part of a non-homologous chromosome moves to another chromosome, and this other chromosome also reciprocates the translocation by adding its component to the original chromosome. So let's draw out what this looks like. In a reciprocal translocation, you're going to end up with, for instance, on this bottom one, this smaller one, you'll have I, J genes, and then you'll have A, B from this one. Then this other chromosome is going to get GH, this one, in this reciprocal trait, so to speak, and then C, D, E, and F. So again, a non-reciprocal, you only involve one chromosome. Well, only one chromosome gets the new information. The others just loses it. It doesn't. In this case, this larger black chromosome here doesn't reciprocate and add DNA to this chromosome here. The reciprocal one, however, there's an even swap. Some of the chromosome from one goes to the other, and then vice versa. All right, so we just talked about the two kinds of translocations, reciprocal and non-reciprocal. But now I want to spend some time talking about how they can form. I want to begin with talking about what usually prevents translocations. So remember, we have our chromosomes here, and at the end of all of our chromosomes, so we'll make these different colors to show that they are different chromosomes. At the ends of all of our chromosomes, we have telomeres. And we've talked about telomeres before, and their different roles. One of the roles of telomeres that we didn't talk about is how they prevent translocations. Without the telomeres, the ends of these chromosomes are, for lack of a better way of describing it, kind of sticky. Non-telometric ends like to bind to each other. We say non-telometric ends are reactive. So you could form a translocation if, for instance, maybe both of these telomeres broke down. So let's draw this like so. And you could have it breaking down on both ends, but just for simplicity, I'm just going to have it breaking down on one end. now we have these reactive ends. And now they can come together. And you may end up now with a chromosome that looks something like this. These turn out to be not so great for the cell, having these extra large chromosomes, particularly when they're randomly binding with each other. And this is one of the reasons why it's really important that as these telomeres shrink during cell division, that the cells are targeted to die. Because if they don't, we end up with these chromosomes that end up sticking together. And this is one of the reasons that in some cancer cells, we see these very large chromosomal translocations between different chromosomes, actual co-chromosomes binding to each other. Alright, so let's erase this part here, because another way we could get a translocation is if we had a double strand break. The chromosomes break. And when these chromosomes break, as you can imagine, we end up generating lots of these reactive ends. So this is reactive now, and so is this, so is this. Now usually what happens with a double strand break, we have enzymes in our cells that are going to fix this right up and bring them back together, so it's not a problem. The problem comes when you have multiple chromosomes going through these double strand breaks, and then when they start to repair each other, they don't always pair up with the right broken end. So you could end up with a situation like this, where they now translocate to other ends of the chromosomes. They're once again protected by the telomeres, but we could have problems. Again, if we think back to our discussion with inversions, we might not imagine there being any problem in this situation here, because we have the same genomic content, nothing's changed there, but we could still have a position effect for the same reasons we talked about before, or we could have cut the gene, because again these random breaks are random chromosome breaks. These are how translocations can occur, and this is how they can still have an effect by this position effect, or depending on where the breaks occur. Now what I just drew over there on the previous slide was something like this. I didn't write letters of genes here, but I could have just as easily. But the point I want to make here is that we haven't changed the content of the chromosome. It still has the same number of genes. We said that on the last slide, but I want to give this a name now. We call these translocations, like here, balanced translocations, because the number of genes has not changed, so they're balanced. And typically, unless we have those position effects or those causes because of the randomness of the chromosome breaks, usually these individuals are healthy. If by chance you get a unbalanced translocation, these are usually not healthy and often lethal. So anywhere from not healthy to lethal. Because an unbalanced translocation means that there's part of that genetic material that is now lost. One thing about these balanced translocations, even though the individual is healthy, they very frequently have fertility problems. And this has to do with how the chromosomes align during metaphase and then are segregated during anaphase. Because even though the chromosomal content overall is balanced, each of these chromosomes may have a slight different arrangement, and so now they can get sorted a little differently, and often that will lead to fertility problems. For this, I just want you to know that it can lead to a fertility problem. The actual genetic nature of those fertility problems we will not cover. And that is the most common kind of translocation, and we call these Robertsonian translocations. This is when you take two acrocentric centromeres, and I'm going to use the most common example, and that is chromosomes 14 and 21 in humans. So let's draw chromosome 14 here. It's acrocentric, so that means we have a little bit of a chromosome on top and a large portion on the bottom. And again, this is chromosome 14. And let's draw the other one, chromosome 21, also acrocentric. And so these Robertsonian translocations can occur between any two acrocentric centromeres, but this is just a specific example here. Whenever these translocations occur, they always lead to a metacentric centromere. So it's going to include this part, and then it's going to include this part here from 21. So this one chromosome has 14 and 21. Then you also end up with this other really puny chromosome. It's also metacentric, and it is almost always lost, and that's okay. What you really want to make sure is that you have a copy of 21 and 14. Now remember, in these individuals, so if I were to draw this cell of somebody that is in each of these cases, let's go and do that. Remember, this person has 2 copies of 21, even though I only drew the one originally. It has 2 copies of 14. When this translocation occurs, and I'm going to just erase this guy here, it still has this other copy of 14, this individual does, and this other copy of 21. So this person here is healthy. 2 copies of 14, 2 copies of 21. Overall, they have 1 fewer chromosomes because they don't have 2 specific each of these, but they have the same genetic material and they're healthy. So the problem occurs during gamete development. And we're going to erase this side over here. And I want to talk about meiosis, but I'm not going to go into great detail. Meiosis 1 and meiosis 2. Remember, it's going to package the chromosomes up because they're going to segregate during both of these division states. You could get a gamete now that has just this one in it. You could get a gamete that has this in it. You could also get a gamete that gets this chromosome here, the translocated one, plus one of these. Then we'll draw the third option here. You could also get this chromosome, the translocated chromosome, and 21 by itself. Okay, so I'm going to put T here because that's a translocated one, and this one here has 21 and 14. This one has 21 and 21 and 14. This one has 21, 1 copy of 21, and 2 copies of 14. Alright, the one last thing I want to talk about with translocations is just a brief discussion of the evidence of them occurring throughout evolution. As we drew on a previous slide, we know that humans are here, and then we have chimpanzees here, gorillas here, and other primates. And we know that, for instance, humans and chimpanzees shared a common ancestor here, humans, chimpanzees, gorillas shared a common ancestor here, and down the line. And we know that throughout these branch points, the branch points that went on to become gorillas or the one that went on to become chimpanzees or humans, that different selective pressures existed and that led to different kinds of genetic mutations. All I want you to know about evolution in regards to translocations is that our chromosome 2, which I'm going to draw like so, at one time was likely 2 separate chromosomes. That underwent a type of Robertsonian translocation where these 2 were translocated to give us this. And how do we know that? Or why do we suspect that? Well, we know in chimpanzees, gorillas, and orangutans that the genes on, say, the chimpanzee's chromosomes, on this chromosome, which we now call chromosome 2A, this one we call 2B, homologous genes here on the human chromosome 2. And on the second, what we call 2B in chimpanzees and other primates, we see those exact same genes here. When we talk about banding patterns within chromosomes, that we saw before, we see the exact same banding pattern, or I shouldn't say the exact same, a very similar banding pattern between humans, chromosome 2, and the 2 separate chromosomes of the chimpanzees. We also see evidence of some centromeric linkage sequences here. It's not a true centromere, but it's similar. Did this kind of translocation lead to any selective advantages for humans as we evolved? It's not known, but we could still see some of those position effects, as well as cutting genes in certain ways that may have led to some sort of advantage. Okay, let's erase this and move on to our translocation summary. What you should know about translocations, or should have got from this podcast, is you should know how to draw one and be able to distinguish between a non-reciprocal and a reciprocal. You should know the difference between a balanced and an unbalanced as well. You should know why that people with balanced translocations are healthy, but also they are often have fertility problems. You don't have to be able to map this out and draw the chromosomes moving around. You just need to know in general that people with balanced translocations will sometimes segregate the chromosomes in such a way that it leads to inviable gametes. We spent a good deal of time talking about Robertsonian translocations. And how that can lead to a form of inherited Down syndrome. And then we closed with a special case of Robertsonian translocations in evolution, and how that has allowed, and there's evidence for this, that R1 chromosome 2 at one time used to be 2 separate chromosomes that went under a translocation event to generate this one single chromosome. Alright, that's all I have for this podcast. If you have any questions, please let me know. Thank you. Bye.